Fossil Insects, A Journey to the Past. A presentation produced for the Mississippi State Insect Fair by staff of the Dunn-Seiler Museum in the Department of Geosciences at Mississippi State University. Hosted by Amy Moe-Hoffman, curator of the Dunn-Seiler Museum. Hi everyone, and welcome to the Insect Fair. My name is Amy Moe-Hoffman, and I'm the curator of the Dunn-Seiler Museum in the Department of Geosciences at Mississippi State University. People in my department study everything about the Earth, including rocks, weather, the people who live here, and even neighboring planets like Mars. But I am a paleontologist, so I study fossils and past life on the planet. When you think about fossils, what kinds of creatures come to mind? Most likely, you think about big, exciting things like dinosaurs or mastodons, or maybe even fish, because those are the things that get the most attention in the news and maybe in the books in your library at school. I don't study any of those things. I chose to study much cooler fossils. Insects. You might think that insects are too fragile to be preserved, but their chitinous exoskeletons are actually quite strong, and they can be preserved beautifully. The first insect you see here is a lacewing insect, and we saw it at the beginning of the slideshow as well. On the right, you see its living relative, the green lacewing. On the next slide, we see another fossil with a big fly front and center. Look carefully at the bottom right corner and you see a second insect, a parasitoid wasp. Both of these insect fossils are from the Florissant Formation in Colorado and are 34.1 million years old. Studying fossil insects has given me the opportunity to enjoy both fieldwork and laboratory aspects of scientific research. Some days I get to spend time outside searching for fossils and studying the rocks that contain them. Here on the right is a picture of my friend Amanda working with me on a stratigraphic column at a fossil site in Nevada. A stratigraphic column shows us all of the layers of rock at an outcrop, with the oldest layers at the bottom and the youngest layers at the top. On the left is a picture of my daughter working with me here in Mississippi as I look for fossil insects and plants. My research has also allowed me to work in the laboratory on existing fossil collections. Here I am working on identifying and preserving historic Florissant fossil collections, like those we saw in the previous slide. These were at the University of Colorado Museum of Natural History in Boulder, Colorado, and the specimens were collected between 1880 and 1910. Working on museum collections allowed me to rediscover long-hidden fossil treasures and to learn about the history of how those fossils were collected. Reinvestigating existing museum specimens leads to many new discoveries as technologies improve each year. This third photo shows my graduate school advisor, Dr. Dina Smith, and a fellow student looking at some of the amazing fossils we studied at our lab in Colorado. Now let's talk for a moment about how insects are preserved as fossils. There are lots of ways they can be preserved, but some forms of insect fossilization are quite rare, like these scarab beetles that have been preserved in the La Brea tar pits in California. Or like these that have been replaced by other minerals like silica quartz or calcite. This is a dragonfly nymph. Others show evidence of insect life habits, like these nests and tunnels. These are called trace fossils. Much more common, however, are one of these three types of fossil preservation, which I'm going to focus on today. 1, we have compression fossils. 2, insects preserved in amber. And 3, a special type of trace fossil that deals with insect-plant interactions. These might be leaves that show us evidence of insects eating them or laying eggs in them. We're going to start with compression fossils. The rocks that contain compression fossils are fine-grained sedimentary rocks called shales, which form in the bottoms of lakes, ponds, or even in the ocean. Shales form in bodies of water because tiny sediments or pieces of silt and clay have time to sink to the bottom in the still water. Let's start by watching a short video on how insects would be preserved in the shale at the bottom of a lake. It is a beautiful day 34.1 million years ago. We see a beautiful lake surrounded by forest with a majestic mountain in the backdrop. A dragonfly is minding its own business, flying across the lake looking for little midges or other insects to eat. Something goes wrong and the dragonfly dies, falling into the water. At first the dragonfly floats on top of the water, but after a bit of time he becomes waterlogged and he begins to fall to the bottom of the lake. But before the dragonfly is decomposed by bacteria, something really exciting happens. The majestic mountain in the backdrop is actually a volcano, and it begins to erupt, shooting tons of ash into the atmosphere. The ash travels across the land and over the lake, settling down through the water to be deposited at the bottom of the lake. Over time, more sediment is deposited in the lake, further covering the dead insect. Sediments can come from erosion of the now extinct volcano, or from flood events, or just from normal stream flow into the lake basin. The weight of the new sediment compresses the layers below, lithifying them or turning them into stone, with the insect preserved inside. Eventually, over millions of years, the landscape changes and the volcanoes are gone. The layers of rock are the only evidence we have that the lake once existed there, and the insects and other fossils inside can be studied and read like the pages of a history book. Let's say we're out in the field collecting. You come across a piece of shale that might have fossils hidden between its bedding planes or layers. To open the rock, you would turn it on its side and carefully hit it with a rock hammer or a chisel called a persuader. Always use safety glasses when hitting a rock with a rock hammer. Once you've cracked open the rock, you carefully peel the two sides apart, making sure not to scrape the two sides against one another. The really cool thing about compression fossils is that when an organism is preserved inside, it is preserved on both sides of the rock. This is called a part and counterpart. You always want to keep the part and counterpart together in a collection because different features might be preserved on one side of the rock than the other. In this rock, you can clearly see the mirrored image of a wetland grass in part and counterpart. And here we can see a close-up photo of a part and counterpart of a Lower Cretaceous beetle in the Copetidae family, approximately 120 million years old. The part on the right shows the dorsal or back side of the beetle with its hard outer wings called elytra and its eyes up toward the top of the photo. The counterpart on the left shows the ventral or underside of the beetle, so there we see very clearly the insect's legs, thorax, mouth parts, and antennae. Before the importance of insect fossils was fully realized, many museums would send their counterparts to other museums, which means that a lot of scientific value of these specimens were lost. Finally, here is what a modern-day relative of this beetle looks like. One way that scientists can better study fossils is to look at them under a stereo microscope, just like in this picture from earlier in the presentation. Or, if you're in the field working without a microscope, you can use a jeweler's lens, also called a loupe lens. I wish you could take a look at some of our specimens under a real microscope today, but since we aren't able to do that, I'm going to zoom in on a few of our insects so you can get an idea of how different they look when they're magnified. Here is a large slab of Green River shale, one of the recent donations to our museum. The Green River Formation is found in many western states, including Colorado, Utah, and Wyoming. These specimens are early Eocene in age, approximately 50 million years old, and as we zoom in, we see some beautiful insects coming into focus. Front and center is a hemipteran, a true bug also known as a stink bug. Take a closer look. Do you think that this side of the fossil is showing the dorsal view, the back side of the insect? Or do you think it's showing the ventral view, the belly view of the insect? If you said the fossil showed the ventral or belly view of the hemipteran, you were right. Now let's zoom in on a really great beetle. This specimen is also one of our recently donated insects from the Green River Formation. You can see his thick protective outer wings very clearly, which means that this side of the fossil is its dorsal view. Do you remember what the beetle's hard outer wings are called? It's okay if you don't remember. I said it very quickly earlier in the video. They are called elytra. Fossil insects have been studied for hundreds of years. This is a page from a monograph or scientific report written by a researcher named Samuel Scudder back in 1890. Even though it's a really old book, the images and research completed by Scudder are still very important references for fossil insects today. Can you find our fossil beetle here on this page of Scudder's book? It's right here, outlined by the circle. Good job. Finally, here's an image of a recent relative of this particular fossil insect, along with another fossil that shows off some of their really fantastic eyes. Now we move on to my favorite and the focus of my graduate research, the flies, or Diptera. This is a bibionid fly, also known as a march fly. You may have seen these guys around your house in Mississippi. We call them love bugs, and they look like this. Notice that this specimen looks a little different than the other fossils for a couple of reasons. First, it isn't preserved in dorsal or ventral view, but actually on its side in what we call lateral view, like this recent bibionid fly. When I was studying fossil flies, it was really common for this genus to be preserved on its side while all of the other flies were preserved showing either the dorsal or ventral views. There is an entire field of fossil study called taphonomy, where researchers try to answer questions like how and why an organism is preserved the way it is, or to answer questions like I had about these plesia. Why are they preserved on their sides when other insects aren't? Now let's switch gears. Do you remember how many wings an insect has? If you said 4, you're correct. But how many wings do you see in our fossil here? If you said 2, you're also correct. Remember that flies have 2 wings. In this image, our fly on the left has 2 wings, and the area highlighted in pink on his body are where the former second wings were. They were, over time, replaced by little stubs called halteres. So our little fly friends have 2 wings instead of 4. Let's transition now to insects preserved in amber. Amber is a very sticky resin or pitch from a tree. As resin comes out of a tree, insects, leaves, pollen, and even vertebrates like lizards or birds can get stuck and covered. Over time, the stuck creature dries out, kind of like a mummy. You may be familiar with this type of fossil from the movie Jurassic Park, which is about a theme park that cloned dinosaurs from DNA removed from a fossil mosquito preserved in amber. Just to be clear, DNA has never been recovered from an insect preserved in amber, so you don't need to worry about anyone cloning dinosaurs or dinosaurs running around terrorizing the world anytime soon. Insects preserved in amber are essentially frozen in 3 dimensions. Sometimes many insects are preserved together, but sometimes a single insect is found. But no matter what, the details you can see are really astounding. Even the smallest details can be seen in the insect, from its mouth parts to its wing veins to even the smallest hairs that are on its body. Finally, we get to plant-insect interactions. Fossils that show how plants and insects interact can tell us a lot about the environment and climate that existed where those insects lived, and how plants and insects have changed together through time. This red line shows a leaf mine, which happens when an insect lays an egg inside of a leaf. The egg hatches and the offspring start off their lives by eating through the leaf in this curvy sort of pattern until they're big enough to find food on their own. We also see some chew marks at the top edge of the leaf, made by an adult insect. A lot of times, the damage that an insect makes on a leaf is specific to the genus or species, and it isn't always directly related to food. For instance, look at this neat circular damage to a recent leaf caused by a leafcutter bee. This same damage can be seen in fluorescent fossil leaves from 34 million years ago. This interaction between the leafcutter and this particular type of plant, the salvia, has persisted across that entire time. Thanks for joining us today to learn more about fossil insects at the Insect Fair. We hope you enjoyed the program. This video was produced and recorded for the Insect Fair by Amy Moe-Hoffman, curator of the Dunn-Seiler Museum, with the help of Dr. Renee Clary. A special thanks to all of the organizers for this event, and we hope to see you all in person at the museum someday soon. Bye, everybody.